Hey everyone, welcome to NCERT Audiobook Unit 14 Biomolecules. It is the harmonious and synchronous process of chemical reactions in body which leads to life. A living system grows, sustains, and reproduces itself. The most amazing thing about a living system is that it is composed of non-living atoms and molecules. The pursuit of knowledge of what goes on chemically within a living system falls in the domain of biochemistry. Living systems are made up of various complex biomolecules like carbohydrates, proteins, nucleic acids, lipids, etc. Proteins and carbohydrates are essential constituents of our food. These biomolecules interact with each other and constitute the molecular logic of life processes. In addition, some simple molecules like vitamins and mineral salts also play an important role in the functions of organisms. The structures and functions of some of these biomolecules are discussed in this unit. Carbohydrate 14.1 Carbohydrates. Carbohydrates are primarily produced by plants and form a very large group of naturally occurring organic compounds. Some common examples of carbohydrates are cane sugar, glucose, starch, etc. Most of them have a general formula 6xH2OY and were considered as hydrates of carbon, from where the name carbohydrate was derived. For example, the molecular formula of glucose C6H12O6 fits into this formula. C6H2O whole 6. But all the compounds which fit into this formula may not be classified as carbohydrates. For example, acetic acid CH3COOH fits into this general formula C2H2O whole 2 but is not a carbohydrate. Similarly, ramenose C6H12O5 is a carbohydrate but does not fit in this definition. A large number of their reactions have shown that they contain a specific functional groups. Chemically, the carbohydrates may be defined as optically active polyhydroxyaldehydes or ketones, or the compounds which produce such units on hydrolysis. Some of the carbohydrates which are sweet in taste are also called sugars. The most common sugar used in our homes is named as sucrose, where the sugar present in milk is known as lactose. Carbohydrates are also called saccharides. Greek saccharon means sugar. 14.1.1 Classification of carbohydrates. Carbohydrates are classified on the basis of their behavior on hydrolysis. They have been broadly divided into following three groups. 1. Monosaccharides, a carbohydrate that cannot be hydrolyzed further to give simpler unit of polyhydroxyaldehyde or ketone is called a monosaccharide. About 20 monosaccharides are known to occur in nature. Some common examples are glucose, fructose, ribose, etc. Second, oligosaccharides. Carbohydrates that yield 2 to 10 monosaccharide units on hydrolysis are called oligosaccharides. They are further classified as disaccharides, trisaccharides, tetrasaccharides, etc., depending upon the number of monosaccharides they provide on hydrolysis. Amongst these, the most common are disaccharides. The two monosaccharide units obtained on hydrolysis of a disaccharide may be same or different. For example, one molecule of sucrose on hydrolysis gives one molecule of glucose and one molecule of fructose, whereas maltose gives two molecules of only glucose. Third, polysaccharides. Carbohydrates which yield a large number of monosaccharide units on hydrolysis are called polysaccharides. Some common examples are starch, cellulose, glycogen, gums, etc. Polysaccharides are not sweet in taste, hence they are also called non-sugars. The carbohydrates may also be classified as either reducing or non-reducing sugars. All those carbohydrates which reduce failing solution and tolerance reagent are referred to as reducing sugars. All monosaccharides, whether aldose or ketose, are reducing sugars. 14.1.2 Monosaccharides. Monosaccharides are further classified on the basis of number of carbon atoms and the functional group present in them. If a monosaccharide contains an aldehyde group, it is known as an aldose, and if it contains a keto group, it is known as ketose. Number of carbon atoms constituting the monosaccharide is also introduced in the name, as is evident from the examples given in table 14.1. Glucose. Glucose occurs freely in nature as well as in the combined form. It is present in sweet fruits and honey. Ripe grapes also contain glucose in large amount. It is prepared as follows. Preparation of glucose. 1. From sucrose, cane sugar. If sucrose is boiled with dilute HCl or H2SO4 in alcoholic solution, glucose and fructose are obtained in equal amounts. 2. From a starch. Commercially, glucose is obtained by hydrolysis of a starch by boiling it with dilute H2SO4 at 393 Kelvin under pressure. A structure of glucose. Glucose is an aldohexose and is also known as dextrose. It is the monomer of many of the larger carbohydrates, namely starch, cellulose. It is probably the most abundant organic compound on earth. It was assigned the structure given below on the basis of the following evidences. 1. Its molecular formula was found to be C6H12O6. 2. On prolonged heating with HI, it forms N-hexane, suggesting that all the 6 carbon atoms are linked in a straight chain. 3. Glucose reacts with hydroxylamine to form an oxime and adds a molecule of hydrogen cyanide to give cyanohydrine. These reactions confirm the presence of a carbonyl group in glucose. 4. Glucose gets oxidized to 6-carbon carboxylic acid, gluconic acid, on reaction with a mild oxidizing agent like bromine water. This indicates that the carbonyl group is present as an aldehydic group. 5. Acetylation of glucose with acetic anhydride gives glucose pentaacetate, which confirms the presence of 5 OH groups. Since it exists as a stable compound, 5 OH groups should be attached to different carbon atoms. 6. On oxidation with nitric acid, glucose as well as gluconic acid both yield a dicarboxylic acid, saccharic acid. This indicates the presence of a primary alcohol OH group in glucose. The exact spatial arrangement of different OH groups was given by Fischer after studying many other properties. Its configuration is correctly represented as 1, so gluconic acid is represented as 2 and saccharic acid as 3. You can see the figure. Glucose is correctly named as D plus glucose. D before the name of glucose represents the configuration, whereas plus represents the extra-rotatory nature of the molecule. It should be remembered that D and L have no relation with the optical activity of the compound. They are also not related to letter D and L. The meaning of D and L notations is as follows. The letters D or L before the name of any compound indicate the relative configuration of a particular stereoisomer of a compound with respect to configuration of some other compound, configuration of which is known. In the case of carbohydrates, this refers to their relation with a particular isomer of glyceraldehyde. Glyceraldehyde contains one asymmetric carbon atom and exists in 2 enantiomeric forms, as shown below. Plus isomer of glyceraldehyde has D configuration. It means that when its structural formula is written on paper following a specific conventions which you will study in higher classes, the OH group lies on right hand side in the structure. All those compounds which can be chemically correlated to D plus isomer of glyceraldehyde are said to have D minus configuration, whereas those which can be correlated to L minus isomer of glyceraldehyde are said to have L configuration. In L minus isomer, OH group is on the left hand side as you can see in the structure. For assigning the configuration of monosaccharides, it is the lowest asymmetric carbon atom which is compared. As in plus glucose, OH on the lowest asymmetric carbon is on the right side, which is comparable to plus glyceraldehyde, so plus glucose is assigned D configuration. Other asymmetric carbon atoms of glucose are not considered for this comparison. Also, the structure of glucose and glyceraldehyde is written in a way that most oxidized carbon, in this case CHO, is at the top. Cyclic structure of glucose. The structure 1 of glucose explained most of its properties, but the following reactions and facts could not be explained by this structure. 1. Despite having the aldehyde group, glucose does not give Schiff's test and it does not form the hydrogen sulfide addition product with NaSO3. Second, the pentaacetate of glucose does not react with hydroxylamine, indicating the absence of free CHO group. 3. Glucose is found to exist in 2 different crystalline forms which are named as alpha and beta. The alpha form of glucose, melting point 419 Kelvin, is obtained by crystallization from concentrated solution of glucose at 303 Kelvin, while the beta form, melting point 400 to 423 Kelvin, is obtained by crystallization from hot and saturated aqueous solution at 371 Kelvin. This behavior could not be explained by the open chain structure for glucose. It was proposed that one of the OH groups may add to the CHO group and form a cyclic hemiacetal structure. It was found that glucose forms a six-membered ring in which OH at C5 is involved in ring formation. This explains the absence of CHO group and also existence of glucose in two forms as shown below. These two cyclic forms exist in equilibrium with open chain structure. The two cyclic hemiacetal forms of glucose differ only in the configuration of the hydroxyl group at C1 called anomeric carbon. The aldehyde carbon before cyclization, such isomers that is alpha form and beta form are called anomers. The six-membered cyclic structure of glucose is called pyranose structure, alpha or beta, in analogy with pyran. Pyran is a cyclic organic compound with one oxygen atom and five carbon atoms in the ring. The cyclic structure of glucose is more correctly represented by Haworth structure as given below. Fructose. Fructose is an important ketohexose. It is obtained along with glucose by the hydrolysis of disaccharide sucrose. It is a natural monosaccharide found in fruits, honey, and vegetables. In its pure form, it is used as a sweetener. It is also an important ketohexose. A structure of fructose. Fructose also has the molecular formula C6H12O6, and on the basis of its reactions, it was found to contain a ketonic functional group at carbon number 2 and 6 carbons in a straight chain, as in the case of glucose. It belongs to D series and is a laboratory compound. It is approximately written as D minus fructose. Its open chain structure is as shown. It also exists in two cyclic forms which are obtained D minus fructose by the addition of OH at C5 to the carbonyl group. The ring thus formed is a five-membered ring and is named as furanose with analogy to the compound furan. Furan is a five-membered cyclic compound with one oxygen and four carbon atoms. The cyclic structures of two anomers of fructose are represented by Haworth structures. Disaccharides. You have already read that disaccharides on hydrolysis with dilute acids or enzymes yield two molecules of either the same or different monosaccharides. The two monosaccharides are joined together by an oxide linkage formed by the loss of a water molecule. Such a linkage between two monosaccharide units through oxygen atom is called glycosidic linkage. In disaccharides, if the reducing groups of monosaccharides, that is aldehydic or ketonic groups, are bonded, these are non-reducing sugars. Example, sucrose. On the other hand, sugars in which these functional groups are free are called reducing sugars. For example, maltose and galactose. Sucrose. One of the common disaccharides is sucrose, which on hydrolysis gives equimolar mixture of D plus glucose and D minus fructose. These two monosaccharides are held together by a glycosidic linkage between C1 of alpha-D-glucose and C2 of beta-D-fructose. Since the reducing groups of glucose and fructose are involved in glycosidic bond formation, sucrose is a non-reducing sugar. Sucrose is dextrorotatory but after hydrolysis gives dextrorotatory glucose and levorotatory fructose. Since the levorotation of fructose minus 92.4 degree is more than the dextrorotation of glucose plus 52.5 degree, the mixture is levorotatory. Thus, hydrolysis of sucrose brings about a change in the sign of rotation from dextro plus to levo minus, and the product is named as invert sugar. Second, maltose, another disaccharide. Maltose is composed of two alpha-D-glucose units in which C1 of one glucose is linked to C4 of another glucose unit. The free aldehyde group can be produced at C1 of second glucose in solution and it shows reducing properties, so it is a reducing sugar. Third, lactose. It is more commonly known as milk sugar since this disaccharide is found in milk. It is composed of beta-D-galactose and beta-D-glucose. The linkage is between C1 of galactose and C4 of glucose. Free aldehyde group may be produced at C1 of glucose unit, hence it is a reducing sugar. Polysaccharides. Polysaccharides contain a large number of monosaccharide units joined together by a glycosidic linkages. These are the most commonly encountered carbohydrates in nature. They mainly act as the food storage or structural materials. Starch. Starch is the main storage polysaccharide of plants. It is the most important dietary source for human beings. High content of starch is found in cereals, roots, tubers, and some vegetables. It is a polymer of alpha glucose and consists of two components, amylose and amylopectin. Amylose is water-soluble component which constitutes about 15 to 20% of starch. Chemically, amylose is a long unbranched chain with 200 to 1000 alpha D plus glucose units held together by C1C4 glycosidic linkage. Amylopectin is insoluble in water and constitutes about 80 to 85% of starch. It is a branch and polymer of alpha-D glucose units in which chain is formed by C1C4 glycosidic linkage, whereas branching occurs by C1C6 glycosidic linkage. Second, cellulose. Cellulose occurs exclusively in plants and it is the most abundant organic substance in plant kingdom. It is a predominant constituent of cell wall of plant cells. Cellulose is a straight chain polysaccharide composed only of beta D glucose units which are joined by glycosidic linkage between C1 of one glucose unit and C4 of the next glucose unit. Glycogen. The carbohydrates are stored in animal body as glycogen. It is also known as animal starch because its structure is similar to amylopectin and is rather more highly branched. It is present in liver, muscles, and brain. When the body needs glucose, enzymes break the glycogen down to glucose. Glycogen are also found in yeast and fungi. Importance of carbohydrates. Carbohydrates are essential for life in both plants and animals. They form a major portion of our food. Honey has been used for a long time as an instant source of energy by Vats in Ayurvedic system of medicine. Carbohydrates are used as a storage molecules as a starch in plants and glycogen in animals. Cell wall of bacteria in plants is made up of cellulose. We build furniture, etc. from cellulose in the form of wood and clothe ourselves with cellulose in the form of cotton fiber. They provide raw materials for many important industries like textiles, paper, lacquers, and breweries. Two aldopentoses with D-ribose and two deoxy-D-ribose are present in nucleic acids. Carbohydrates are found in biosystem in combination with many proteins and lipids. 14.2 Proteins. Proteins are the most abundant biomolecules of the living system. Chief sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. They occur in every part of the body and form the fundamental basis of structure and functions of life. They are also required for growth and maintenance of body. The word protein is derived from Greek word proteos, which means primary or of prime importance. All proteins are polymers of alpha amino acids. 14.2.1 Amino acids. Amino acids contain amino NH2 and carboxyl C double bond OH functional group. Depending upon the relative position of amino group with respect to carboxyl group, the amino acids can be classified as alpha, beta, gamma, delta, and so on. Only alpha amino acids are obtained on hydrolysis of proteins. They may contain other functional groups also. All alpha amino acids have trivial names which usually reflect the property of that compound or its source. Glycine is so named since it has sweet taste. In Greek, glykos means sweet, and tyrosine was first obtained from cheese. In Greek, tyros means cheese. Amino acids are generally represented by a three-letter symbol. Sometimes one-letter symbol is also used. Structures of some commonly occurring amino acids along with their three-letter and one-letter symbols are given in table 14.2. Classification of amino acids. Amino acids are classified as acidic, basic, or neutral depending upon the relative number of amino and carboxyl groups in their molecule. Equal number of amino and carboxyl groups makes it neutral. More number of amino than carboxyl group makes it basic, and more carboxyl groups are compared to amino groups makes it acidic. The amino acids which can be synthesized in the body are known as non-essential amino acids. On the other hand, those which cannot be synthesized in the body and must be obtained through diet are known as essential amino acids. Amino acids are usually colorless crystalline solids. These are water-soluble, high-melting solids and behave like salts rather than simple amines or carboxylic acids. This behavior is due to the presence of both acidic carboxyl group and basic amino group in the same molecule. In aqueous solution, the carboxyl group can lose a proton and amino group can accept a proton, giving rise to a dipolar ion known as Zwitter ion. This is neutral but contains both positive and negative charges. In Zwitter ionic form, amino acids show amphoteric behavior as they react both with acids and bases. Except glycine, all other naturally occurring alpha amino acids are optically active. Since the alpha carbon atom is asymmetric, these exist both in D and L forms. Most naturally occurring amino acids have L configuration. L amino acids are represented by writing the NH2 group on left hand side. A structure of proteins. You have already read that proteins are the polymers of alpha amino acids and they are connected to each other by peptide bond or peptide linkage. Chemically, peptide linkage is an amide formed between C double bond OH group and NH2 group. The reaction between two molecules of similar or different amino acids proceeds through the combination of the amino group of one molecule with the carboxyl group of the other. This results in the elimination of water molecule and formation of a peptide bond CONH. The product of the reaction is called a dipeptide because it is made up of two amino acids. For example, when carboxyl group of glycine combines with the amino group of alanine, we get a dipeptide glycylalanine. If a third amino acid combines to a dipeptide, the product is called a tripeptide. A tripeptide contains three amino acids linked by two peptide linkages. Similarly, when 4, 5 or 6 amino acids are linked, the respective products are known as tetrapeptide, pentapeptide or hexapeptide, respectively. When the number of such amino acids is more than 10, then the products are called polypeptides. A polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 U is called a protein. However, the distinction between a polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids are likely to be called proteins if they ordinarily have a well-defined conformation of a protein, such as insulin, which contains 51 amino acids. Proteins can be classified into two types on the basis of their molecular shape. A. Fibrous proteins. When the polypeptide chains run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. Some common examples are keratin, present in hair, wool, silk, and myosin, present in muscles. B. Globular proteins. This structure results when the chains of polypeptide coil around to give a spherical shape. These are usually soluble in water. Insulin and albumins are the common examples of globular proteins. The structure and shape of proteins can be studied at four different levels, that is primary, secondary, tertiary and quaternary, each level being more complex than the previous one. Primary structure of proteins. Proteins may have one or more polypeptide chains. Each polypeptide in a protein has amino acids linked with each other in a specific sequence, and it is this sequence of amino acid that is said to be the primary structure of that protein. Any change in this primary structure, that is sequence of amino acid, creates a different protein. Secondary structure of proteins. The secondary structure of protein refers to the shape in which a long polypeptide chain can exist. They are found to exist in two different types of structures, with alpha helix and beta pleated sheet structure. These structures arise due to the regular folding of backbone of polypeptide chain due to hydrogen bonding between carbonyl, C double bond O and NH group of the peptide bond. Alpha helix is one of the most common ways in which a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw helix with the NH group of each amino acid residue hydrogen bonded to carbonyl of an adjacent turn of the helix. In beta pleated sheet structure, all peptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonds. The structure resembles the pleated folds of drapery and therefore is known as beta pleated sheet. Tertiary structure of proteins. The tertiary structure of proteins represents overall folding of the polypeptide chains, that is, further folding of the secondary structure. It gives rise to two major molecular shapes, fibrous and globular. The main forces which stabilize the secondary and tertiary structure of proteins are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic forces of attraction. Fourth, quaternary structure of proteins. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. A diagrammatic representation of all these four structures is given in figure 14.3, where each colored ball represents an amino acid. The given figure is here. Denaturation of proteins. Protein found in a biological system with a unique three-dimensional structure and biological activity is called a native protein. When a protein in its native form is subjected to physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed. Due to this, globules unfold and helix get uncoiled and protein loses its biological activity. This is called denaturation of protein. During denaturation, secondary and tertiary structures are destroyed, but primary structure remains intact. The coagulation of egg white on boiling is a common example of denaturation. Another example is curdling of milk, which is caused due to the formation of lactic acid by the bacteria present in milk. 14.3 Enzymes. Life is possible due to the coordination of various chemical reactions in living organisms. An example is the digestion of food, absorption of appropriate molecules, and ultimately production of energy. This process involves a sequence of reactions and all these reactions occur in body under very mild conditions. This occurs with the help of certain biocatalysts called enzymes. Almost all the enzymes are globular proteins. Enzymes are very specific for a particular reaction and for a particular substrate. They are generally named after the compound or class of compounds upon which they work. For example, the enzyme that catalyzes hydrolysis of maltose into glucose is named as maltase. Sometimes enzymes are also named after the reaction where they are used. For example, the enzymes which catalyze the oxidation of one substrate with simultaneous reduction of another substrate are named as oxidoreductase enzymes. The ending of the name of an enzyme is ase. Mechanism of enzyme action. Enzymes are needed only in small quantities for the progress of a reaction. Similar to the action of chemical catalyst, enzymes are said to reduce the magnitude of activation energy. For example, activation energy for acid hydrolysis of sucrose is 6.22 kJ per mole, while the activation energy is only 2.15 kJ per mole when hydrolyzed by the enzyme sucrase. Mechanism for the enzyme action has been discussed in Unit 5. 14.4 Vitamins. It has been observed that certain organic compounds are required in small amounts in our diet, but the deficiency causes specific diseases. These compounds are called vitamins. Most of the vitamins cannot be synthesized in our body, but plants can synthesize almost all of them, so they are considered as essential food factors. However, the bacteria of the gut can produce some of the vitamins required by us. All the vitamins are generally available in our diet. Different vitamins belong to various chemical classes and it is difficult to define them on the basis of a structure. They are generally regarded as organic compounds required in the diet in small amounts to perform specific biological functions for normal maintenance of optimum growth and health of the organism. Vitamins are designed by alphabets A, B, C, D, etc. Some of them are further named as subgroups B1, B2, B6, B12. Excess of vitamins is also harmful and vitamin pills should not be taken without the advice of doctor. The term vitamine was coined from the word vital plus amine since the earlier identified compounds had amino groups. Later work showed that most of them did not contain amino groups, so the letter E was dropped and the term vitamin is used these days. Classification of vitamins. Vitamins are classified into two groups depending upon their solubility in water or fat. First, fat-soluble vitamins. Vitamins which are soluble in fat and oils but insoluble in water are kept in this group. These vitamins A, D, E and K. They are stored in liver and adipose fat-storing tissues. Second, water-soluble vitamins. B group vitamins and vitamin C are soluble in water, so they are grouped together. Water-soluble vitamins must be supplied regularly in diet because they are readily excreted in urine and cannot be stored except vitamin B12 in our body. Some important vitamins, their sources and diseases caused by deficiency are listed in table 14.3. Table 14.3. Vitamin A, source fish liver oil, carrots, butter and milk. Deficiency disease, xerophthalmia, hardening of cornea of eye, night blindness. Vitamin B1, thiamine, yeast, milk, green vegetables and cereals. Deficiency disease, beriberi, loss of appetite, retarded growth. Vitamin B12, riboflavin, sources milk, egg white, liver, kidney. Deficiency disease, cheilosis, fissuring at the corners of mouth and lips, digestive disorders and burning sensation of the skin. Vitamin B6, pyridoxine, source yeast, milk, egg, yolk, cereals and grams. Deficiency disease, convulsions. Vitamin B12, source meat, fish, egg and curd. Deficiency disease, pernicious anemia, RBC deficient in hemoglobin. Vitamin C, ascorbic acid. Source, citrus fruits, amla and green leafy vegetables. Deficiency disease, scurvy, bleeding gums. Vitamin D, exposure to sunlight, fish and egg yolk. Rickets, bone deformities in children, and osteomalacia, soft bones and joint pain in adults. Vitamin E, source vegetable oils like wheat germ oil, sunflower oil, etc. Deficiency disease, increased fragility of RBC and muscular weakness. Vitamin K, source green leafy vegetables. Deficiency disease, increased blood clotting time. Nucleic acid. Every generation of each and every species resembles its ancestors in many ways. How are these characteristics transmitted from one generation to the next? It has been observed that nucleus of a living cell is responsible for this transmission of inherent characters, also called heredity. The particles in nucleus of the cell responsible for heredity are called chromosomes, which are made of proteins and another type of biomolecules called nucleic acid. These are mainly of two types, the deoxyribonucleic acid DNA and ribonucleic acid RNA. Since nucleic acids are long-chain polymers of nucleotides, so they are also called polynucleotides. Chemical composition of nucleic acid. Complete hydrolysis of DNA or RNA yields a pentose sugar, phosphoric acid, and nitrogen containing heterocyclic compounds called bases. In DNA molecules, the sugar moiety is beta D2 deoxyribose, whereas in RNA molecule it is beta D ribose. DNA contains four bases: adenine, guanine, cytosine, and thymine. RNA also contains four bases. The first three bases are same as in DNA, but the fourth one is uracil. A structure of nucleic acid. A unit formed by the attachment of a base to 1' position of sugar is known as nucleoside. In nucleosides, the sugar carbons are numbered as 1', 2', 3', etc. in order to distinguish these from the bases. When nucleoside is linked to phosphoric acid at 5' position of sugar moiety, we get a nucleotide. Nucleotides are joined together by phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar. The formation of a typical dinucleotide is shown in figure 14.6. A simplified version of nucleic acid chain is as shown below. Information regarding the sequence of nucleotides in the chain of a nucleic acid is called its primary structure. Nucleic acids have a secondary structure also. James Watson and Francis Crick gave a double-strand helix structure for DNA. Two nucleic acid chains are wound about each other and held together by hydrogen bond between pairs of bases. The two strands are complementary to each other because the hydrogen bonds are formed between specific pairs of bases. Adenine forms hydrogen bonds with thymine, whereas cytosine forms hydrogen bonds with guanine. In secondary structure of RNA, single-stranded helix is present which sometimes folds back on itself. RNA molecules are of three types and they perform different functions. They are named as messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, tRNA. Biological functions of nucleic acid. DNA is the chemical basis of heredity and may be regarded as the reserve of genetic information. DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. A DNA molecule is capable of self-duplication during cell division, and identical DNA strands are transferred to daughter cells. Another important function of nucleic acid is the protein synthesis in the cell. Actually, the proteins are synthesized by various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in DNA. 14.6 Hormones. Hormones are molecules that act as intercellular messengers. These are produced by endocrine glands in the body and are poured directly in the bloodstream, which transports them to the site of action. In terms of chemical nature, some of these are steroids, example estrogens and androgens. Some are polypeptides, for example insulin and endorphins, and some others are amino acid derivatives such as epinephrine and norepinephrine. Hormones have several functions in the body. They help to maintain the balance of biological activities in the body. The role of insulin in keeping the blood glucose level within the narrow limit is an example of this function. Insulin is released in response to the rapid rise in blood glucose level. On the other hand, hormone glucagon tends to increase the glucose level in the blood. The two hormones together regulate the glucose level in the blood. Epinephrine and norepinephrine mediate responses to external stimuli. Growth hormones and sex hormones play a role in growth and development. Thyroxine produced in thyroid gland is an iodinated derivative of amino acid tyrosine. Abnormally low level of thyroxine leads to hypothyroidism, which is characterized by letharginess and obesity. Increased level of thyroxine causes hyperthyroidism. Low level of iodine in the diet may lead to hypothyroidism and enlargement of the thyroid gland. This condition is largely being controlled by adding sodium iodide to commercial table salt, iodized salt. Steroid hormones are produced by adrenal cortex and gonads, testes in males and ovaries in females. Hormones released by adrenal cortex play a very important role in the functions of the body. For example, glucocorticoids control the carbohydrate metabolism, modulate inflammatory reactions, and are involved in reactions to stress. The mineralocorticoids control the level of excretion of water and salt by the kidney. If adrenal cortex does not function properly, then one of the results may be Addison's disease characterized by hypoglycemia, weakness, and increased susceptibility to stress. The disease is fatal unless it is treated by glucocorticoids and mineralocorticoids. Hormones released by gonads are responsible for development of secondary sex characters. Testosterone is the major sex hormone produced in males. It is responsible for development of secondary male characteristics, deep voice, facial hair, general physical constitution, and estradiol is the main female sex hormone. It is responsible for development of secondary female characteristics and participates in the control of menstrual cycle. Progesterone is responsible for preparing the uterus for implantation of fertilized egg. That's all for today. Thank you.